Hey guys, my name is Max and welcome to Simple Biology. Today we're going to be talking about the endomembrane system, which is actually not as complicated as it sounds. It's basically just a group of organelles that are grouped together because they're very similar in structure. The similarity is that they're made up of membranes and not much else. So the first thing we're going to look at is vesicles. Vesicles are basically just a bubble, a packet of membrane, and it stores proteins and other materials. The reason vesicles are important is because, for example, if a material is in one location and needs to go to another location, the vesicle allows it to go there without the materials just dispersing throughout the cell. And this little picture kind of shows how a vesicle forms. There's a membrane that already exists, and certain processes start to make it bend outward, and then it pinches, and then it starts to pinch. And when it closes off completely, that new little pocket of membrane is a vesicle. And whatever the material was that was in this area is now stored inside the vesicle and can be transported elsewhere. The second organelle we're going to look at is called the Endoplasmic Reticulum. Now I mentioned this briefly when we talked about ribosomes, and so you may know a little bit about this, but what you need to know is that it's continuous with the nuclear membrane, which is right here. We'll talk about the nuclear membrane shortly, but this area that's continuous with it is also called the lumen. And so the area between the membranes of the endoplasmic reticulum is called the lumen. Now there are two parts to the endoplasmic reticulum, the smooth part and the rough part. The difference is that the rough endoplasmic reticulum has ribosomes attached to it, whereas the smooth part does not. Now they have various functions, but the smooth part functions more in synthesizing lipids and some other functions in your liver cells. The smooth endoplasmic reticulum is the part of it that detoxifies certain chemicals, whereas the rough endoplasmic reticulum is actually a little bit more important. This is where the proteins from our bound ribosomes. This is where they are. This is where they first are created, and where they are then first packaged and sorted and sent to other locations. Now, generally, the first place they're going to go is what's called the Golgi Apparatus. And now, this is kind of a picture of a Golgi apparatus. On this end up here, you'll have the endoplasmic reticulum, and on the other end over here is the cell membrane, where the cell ends. If you look at the Golgi apparatus, it structurally looks like a stack of flattened membranes. In some textbooks, it compares them to looking like a stack of pancakes. Yeah. They function in, again, protein sorting, packing, and shipping. Now this is important because as the proteins go through this, they have many different areas where the Golgi apparatus can distinguish between different proteins and determine for them to go to different places where they're going to be doing their function. As the proteins go through each layer, you can see right here, the different vesicles will butt off and fuse with the next layer, generally only in one direction, from the side where the endoplasmic reticulum was to the side where the cell membrane was. And now these two different sides have names. As you can see here, the cis face is the side that faces the endoplasmic reticulum, and the trans face is the side that faces the cell membrane. You can remember this because the trans face is the side where proteins are transported outside of the cell. Moving on, the next organelle is what's called a lysosome. In reality, I think they're a little bit bigger than this relative to other organelles, but they're structurally very similar to vesicles. Now, their function is strongly related to what's located inside them, which are digestive enzymes. And now what they do is these lysosomes will fuse with other with different vesicles, basically, that have stored sometimes food, sometimes damaged organelles. In this picture, it's a chloroplast. We'll be talking about that later. But a membrane will form around this damaged chloroplast or any other organelle or food source for that matter. And the vesicle will go and it'll fuse with that, releasing its contents and allowing that organelle or piece of food to be broken down into its base components, broken down into different sugars, different amino acids, simple lipids. And by doing so, it allows the cell to recycle its contents and stay alive, really. The last thing is what's called a vacuole. Now, a vacuole, there are actually several types of vacuoles. In animals, you have just scattered vacuoles, very small. In plants, they've all combined to form a central vacuole. And in certain other organisms, there's what's called a contractile vacuole. We'll be going over those last two in a minute, but as a basic overview of its structure, a vacuole is basically a large pouch of membrane, and inside of it is just a solution of, it has water, different solutes depending on what needs to be stored there, and it basically functions as a huge vesicle where the cell can store some of its sometimes food or other substances. Like I mentioned, in plants you have central vacuoles, and they have a more important function than just storing stuff in plants. In fact, the contractile vacuoles will pump water in order to allow the cell to grow and expand. Then the other, there's not a picture for this one, but the other type of vacuole is a contractile vacuole, which has the opposite function, and it pumps water out of the cell. This is in certain single-celled eukaryotes. Like if you know what an amoeba is, it has a contractile vacuole, and what the purpose of having a contractile vacuole is that it prevents the cell from absorbing too much water, because just the nature of the chemistry, since there's a higher concentration of solutes inside the cell than outside the cell, water will diffuse into the cell through osmosis. Contractile vacuoles oppose that motion. The endomembrane system is as simple as that.